Indian Institute of Space Science and Technology. Our goal is to bring about a synergy among alumni, students, faculty of IIST for mutual and societal growth. And in this aspect, we've been conducting various sessions, of which today's discussion is the first major event open for anyone interested in space biology. With the upcoming missions of Gaganyaan and PS4 platform being available for microgravity experiments, it offers new set of opportunities for a lot of Indian scientific community in the realm of space biology. It is hence our pleasure to enable a step forward in this direction and to welcome you to today's discussion on space biology. I now invite Dr. Dadwal, Director of IIST, to kindly open this event with the inaugural address. Sir.
Good evening, all participants. Let me specifically thank Prakhar, and his team which took the initiative. Let me also thank Dr. Siddharth and the panel he put, Dr. Sangram, Afshin, and the all others who have joined. Let me start by saying the astrobiology is really a very huge subject, but it has yet to come to a stage so that in the first decade, even IIST neither had an elective nor a research program in this subject. So incidentally, with the help of a couple of our colleagues and Dr. Srija Lakshmi and the collaboration with Dr. Ravi, the IIST has proposed a Drosophila experiment, stone formation in the kidneys, which has been accepted for the first flight of Gaganyaan. Parallelly, Dr. Palash has proposed the gas monitoring with his sensors for the module to see that it is benign for the human. Parallelly, Dr. Selva Ganeshan has now a PhD student who is working on modeling the controls of the human body. But these are just tip of the iceberg. Such a big subject, astrobiology is, not only it touches the human beings, it also touches all the organisms. It also touches all the plants which we need to take, or whether we will go alone to the space, and the human body cannot go without organisms, large number, which are outside and inside us. It also opens a large number of scientific questions from origin of life to whether the life can be seeded from outside, the survivability, and a host of other things, including the science of hypergravity, how the plant development to animal to human body physiology, they react to gravity and also the absence of gravity. So the scope is so large, and we have very, very interested audience. So my suggestion to you is, how will you make this transition from engineering to biology? Obviously, I would recommend, please join with some biologists. It should not be greatly difficult to pick up the subject, and I am quite competent to say so, since I am by training a plant physiologist and I spent last 37 years doing something related to space. So for you also to pick up biology should not be difficult. The second important issue is there are questions of ethics, because we cannot by mistake take organisms outside. And third most important is, since it will be quite costly, delayed, and difficult to really have space experiment, you need to decide what you would do on Earth to really answer the preparedness or the related questions that can be dealt with on the Earth. Since there are very, very specialist panelists and the moderator, Dr. Siddharth, who heads the center at Amity. I'm not sure how many other places in India there is such a focus on astrobiology. So I would say that it gives me great pleasure to inaugurate the event, and I look forward to very, very fruitful discussions, suggestions, and this will lead forward to further experiments and studies from India. Uh, I'm sure large number of Indians do astrobiology, but I would like to see more India-based studies, although science is international, and when you talk of space, it goes even beyond Earth. But I would really like to see more of our friends taking interest in the astrobiology. Thank you very much.
Thank you, sir. Thank you for that inaugural address. Coming over to our main business of the day, we have Dr. Siddharth, who has been very helpful and in collaborating with us for organizing this event today. He is heading the Amity Center for Astrobiology. Him to take over and go ahead with the panel discussion. Dr. Siddharth.
Thank you very much, Prakhar. Am I audible? Okay. First of all, I would like to thank on behalf of Amity University, the Alumni Institution of IIST, Prakhar and your team for pulling off such an event at short notice and being able to give this opportunity. I'm seeing about 195 students are with us. So first of all, thank you for reaching out and connecting with us. We are very, very excited to have more such webinars. And I would also like to pass on my greeting to Dr. Dadwal. And we have a lot to learn from you, sir, because you're having the biology background and you've worked with ISRO for so many years. And I think there are not many people who are having this kind of a background. So maybe next time we will request you as well to, share some notes on your experiences. And it's so nice to have you with us, and your inputs during the Q&A as well will be very useful. So I think I will take the next 5 minutes just sharing some of my thoughts, as well as I have my presentation. So right after my thoughts, we'll, I think maybe one by one, share 10 minutes of our backgrounds and our thoughts. And I think what I will start with is, just like Sir mentioned, astrobiology is a very wide field. And in fact, if you look at some of the other countries, such as in the US, space biology and astrobiology are actually two different areas of work. And there is, at some levels there is an overlap, but in on many occasions there isn't a clear overlap. But being a small community here in India, I think it's a growing community of us who are tackling different aspects of space biology. There's people who have been working with small microorganisms and now trying to identify how are they behaving in the absence of gravity or in excess of gravity. We are looking at the effects of radiation on these organisms. We have people looking at plants, they're looking at fruit flies. So there are so many different aspects of it. And now that we have reached that stage where we are getting an opportunity to conduct experiments in low Earth orbit, it's the right time to have these kind of discussions, both within India as well as with our partners outside of India, and try to have an inclusive environment. We are fortunate being at institutions which are already affiliated with, space projects and space people. but there are so many other organizations. We are the tip of the iceberg right here. And it's important to acknowledge that there are people from different backgrounds, different fields who are doing some very relevant work. So the challenges are, how do we find ways in which we can get people from these different lines of research who have been studying biological sciences for their own specific areas? How do we tap into those groups of people? And similarly for students, just like IIST, we have so many engineering students in India who first become engineers and then later on realize what do they want to do, really. And I can at least talk on my behalf. I'm an aerospace engineer, but today I'm working on space projects. I work with people from sciences. And I'm sure there will be many of us here, many students who would have always had this interest and inclination for space biology. So how can we connect them with their interests? I think that is the main thing. What is the role we play as early career, mid-career, senior level researchers in connecting the aspirations of engineering students with what kind of problems would they like to tackle and solve. And I think this is one thing I personally would really like to see happen. So I think that's some of my initial thoughts. And one by one, I'm sure we'll be hearing from all our panelists. But may I take the occasion, Prakhar, to just give an introduction from my side so that my bit is over and then we can move on?
Yeah, please go ahead.
Awesome. All right, I'll just share my screen. See my screen?
Yes, it's visible.
Okay, thank you. All right, so my name is Siddharth Pandey. Greetings to everybody listening to me in India as well as in other parts of our planet. I'm here in Mumbai, and I'm very, very delighted to speak with you and moderate this exciting session on space biology and what does the 2020s hold for us, the next decade, and what are the learnings that we've had. So I'll give a very quick background of my own research and how did I end up from aerospace engineering into space biology and astrobiology. So I have been very fortunate. I started off as an aerospace engineer. That was my true passion right from the start. As many of aerospace engineering students are, we're really passionate about what we do. And I coincidentally started my education at Amity University, where I'm currently working. And after I graduated, I'm 2010 pass out. I was able to get a position at IIT Bombay for a year, did a research fellowship there, and moved to the Netherlands, got myself a master's degree in space systems engineering. And while I was there, I got an opportunity to join the space biosciences division at NASA Ames. So that was an internship opportunity, which rolled into a research scholarly position where I was working as an engineer, building systems, hardware systems. And from there, once I had finished 2 spaceflight experiments and worked with this very exciting team of people, I then moved to Australia. I was able to get myself a position to do a PhD in understanding how Mars rovers behave in really cold environments. So I was basically doing the thermal design work, the modeling and the experimentation, and designing very thermally efficient systems for Mars rovers. And a part of that was for the ExoMars rover, which is flying in 2 years to Mars. And right after that, and in fact, during my PhD, I was collaborating with several researchers in the US who were part of my previous team at NASA Ames. Many of them were astrobiologists. And coincidentally, I had a meeting with many of them in New Zealand, where we were conducting a field expedition. It was an education research experience called Spaceward Bound. And while I was at that expedition, we conceptualized that we had to have an expedition in India. And that's actually another talk altogether on astrobiology, on how we found ourselves in Ladakh. And that's one of the early Mars analog environments. And we conducted a lot of astrobiology research. But my connection with Amity grew from there. And in 2019, so a year and a half ago, I was offered the position to establish Astrobiology Center, which, as far as we know, is the main center in India, or rather the only center that primarily focuses on astrobiology. There are many people in India who are undertaking astrobiology research, but we have been a center that has been given the mandate to conduct astrobiology. I'll talk about a little bit about my experiments. So as many people are familiar with spaceflight, they are just fascinating projects to be part of. It doesn't matter what you're doing. You could be a manager, you could be a technician, an engineer, or a scientist, but it's something that just you take away from that project and it just stays on with you. And Dr. Ravi Kumar, who's with us, and I'll have the pleasure of introducing to the audience too. He was part of Dr. Sharmila Bhattacharya's team, and I was part of that team. We were both team members of the fruit fly lab who worked at the Space Biosciences division. And we were fortunate to work together on two spaceflight experiments. The one that you're seeing right now on your screen is mainly a payload that was designed to study how fruit flies, so those little insects which Dr. Ravi will be talking about, which you'll find on top of your bananas and other fruits. These are excellent model organisms, and we had flown two different strains of these flies. And to achieve all of this within a very compact space, it was a 1.5 U unit, which roughly is about the size of little smaller than your shoe boxes. And within that, we were able to autonomously observe the flight paths of these flies. So we had sent them with their food and everything, and this was going up to the International Space Station. So we were getting the air and the temperature and everything from the station. But as an engineer, I think this is the first time when I really started respecting biology experiments in space. It's very, very challenging. It comes with its own set of engineering challenges, and living organisms are susceptible to having a certain kind of an environment, which becomes very challenging for engineers to kind of maintain. So there are several different challenges that come to be when you're doing these kind of experiments. And I'll be happy to share links with people who are interested on the two different projects that we were part of. So SpaceX 3 and SpaceX 4 were the two flights that our experiment had flown on. One was the Heart Flies experiment, which flew on SpaceX 3, and the other one was Ames fruit fly experiment. So these were both student experiments that as a student I was able to get introduced to be part of, and that was where I picked up the experience of building flight hardware and what are the different challenges that go into building these kind of systems. I'll give a very quick introduction of my center. I will not go into detail, but it's important for me to do this because there are several students here who might be interested in research opportunities and internships and those kind of things. So with that in mind, I will just give an introduction of what we are currently. So we've been in existence for about 18 months. And as we speak, we currently have three main focus areas that we are working in. The first is early Mars analog fieldwork. So we're basically looking at different parts of India that are geologically and geochemically and from an atmosphere point of view, similar to what early Mars was like when it was a better planet. And Ladakh and Lonar are two regions that we have been studying. And we also are involved in designing and development of lunar drills. So this is something that we are currently undertaking in partnership with the Aerospace Engineering Department at IIT Bombay. But the topic for today is space biology, so I will focus on that. So we have been getting our hands dirty and building some experiments for space flight. And apart from that, we have been forging partnerships. We've been working closely with industry as well as other non-profit organizations to help us conduct science outreach and working closely with students from different disciplines, introducing them to astrobiology. And we are also recently a ISRO outreach node for the Indian Institute of Remote Sensing. So whenever there's a online workshop that they conduct, we actually maintain and register a lot of students from across India, not just from Amity, who are able to undertake these courses that ISRO runs, but we monitor their attendance and answer questions and distribute certificates and those kinds of things. So very quickly, just about our experience with spaceflight as a center. So just last year in September, we were selected we were one out of 10 student teams that was selected to fly a small payload on the ISRO PS4 platform. So a company called Exceed Space, they were selected by ISRO to fly a unit that could fly, that had 10 slots that could fly experiments. And we were one out of those 10 teams that got selected for this. And we were basically given 3 months to come up with a piece of hardware and deliver it to them. And that would be flying on the PSLV C-49. So we got ourselves together, and there's a new center I collaborated within my university with the biotechnology school. And the biotechnology school had Dr. Pamela Jha and Dr. Renata Joby. And these two scientists were basically interested in seeing the effect of microgravity on the growth of plant callus. So one of the constraints I think that we faced is whatever living organism that we pack up in through this hardware and give it to ISRO, it would have to survive about 2 months of shelf waiting period. So it's not like we give it to ISRO, for example, on Monday, and then on Friday it's going to space. So normally a launch is a very complex mission. It has several different components. And unlike other kinds of payloads, a biological payload is very time critical. So one thing that is very important for teams that work on such experiments, they really have to ensure that once you've given them your experiment, it has to actually last that entire wait period before it even reaches spaceflight. Because what can happen is that that plant or that organism can in fact use all of its resources and in fact die, and your experiment will be over before it even reaches space. So we actually compared and studied different kinds of living organisms that we can study. And we picked up something that had a slow incubation period that could survive that wait period of about 6 weeks to 8 weeks before it actually got to space. And this experiment was a technology demonstration, but at the same time, it was very challenging. From a technical point of view, it was very small and narrow. So this entire thing roughly fitted about those old school video cassette VHS tapes that you have. So 16 by 10 by 3 centimeters was a slot that we were given. It had to fit within 200 grams. And because of its because of the living organism, the plant, it had to have a very tight thermal limit, so 20 to 35 degrees within which it had to be. And keep in mind, this is on the PS4, not the Gaganyaan. So PS4 is a completely external platform, so it had to be self-contained, vacuum sealed, and so there were all of these different things, and we were limited in terms of the downlink as well. So we only had one pass or maximum 2 passes per day that would be flying over India. And everything had to be downloaded within 255 bytes per pass. So these were the extreme severe engineering challenges, and within 3 months this had to be built. So it was quite a bit of a nightmare. But I was very happy and proud to see that my students at Amity got together and worked overnight for 2.5 months, and we were able to put something together. So an onboard computer would process the images from a camera and send them back to us. So this current experiment is on hold because we haven't been given a launch date. ISRO PSLV C-49 still hasn't got a launch date. That basically means that the plant by now is probably dead and we have to replace it. Those are the kind of risks you take and the different things, unforeseen circumstances that come with spaceflight. And another thing to point out is this was an important collaboration with Paris Systems as well. So that's another thing to keep in mind. So in the interest of time, I will not go into other details. Perhaps we can have a discussion on astrobiology in general, but I've already mentioned that Ladakh and Lonar are the 2 craters that we work on. And we also undertake space biology and astrobiology workshops. So for students who are interested, we do have research opportunities here. So I welcome students to check out our website and also send us an email if they are interested here. And I'm happy to talk about it more later when it comes up. But I would like to move on and would like to stop sharing my screen now. And let's see, perhaps we can have Dr. Afshin Khan, who's joining us from California. So over to you, Dr. Afshin.
All right, thank you, Siddharth. And let's see if, are you looking at the correct screen? Like it's supposed to be a PowerPoint.
Yep, just go full screen.
Yeah, so first I would really like to thank everyone, IIST alumni and Siddharth and everyone else who was involved in setting this panel together. I'm really excited to see so many enthusiastic questions. I was looking at the chat line and seeing so many enthusiastic students. But that's always great. That happens sometimes when we are hiring interns for the Blue Marble Space Summer Program. There's always very enthusiastic students from India, and we get great applications from there. So yeah, we'll talk about the internships. I saw your questions on internships and Mars and other things, so I'll be answering some of those questions. So I am a biologist, and yes, in the chat window, somebody asked, will there be a discussion on how biologists can become astrobiologists? So yes, I am a biologist. During my undergrad, I had a major in biotechnology with minors in genetics and biochemistry. And that's how I got introduced to the field of astrobiology. But this is back in 2006, 2007, when I was in my second year. There weren't any colleges in India, unfortunately, that had a focus in astrobiology. And to be honest, my professors did not know what this was. So I took the initiative and I started attending lectures online by NASA and trying to teach myself what this was all about. And I wrote the first article for my college in Bangalore on what space biology was and how the future of biosciences will be defined by a lot of research that's done in space. So yeah, it was a long journey from there, from an undergrad to now. And eventually I ended up learning a lot of geology and planetary science and geochemistry during my PhD. And now I think I can call myself an astrobiologist, but then when you have trained in so many things, it's kind of hard to describe your background. So yeah, my areas of interest is that I like to study Earth and life systems as an entire system science, and not a specific thing inside of that, but looking at the entire system and how it interacts with its surrounding. And that's something that astrobiologists have to do quite often, especially when they're looking for signs of life on other planets or in different star systems, and depending on what their background is, whether it's astronomy, astrophysics, or biosciences. But yes, biologists can absolutely become astrobiologists. And most of the astrobiologists I work with are in training microbiologists or molecular biologists or plant biologists and other types of biologists. So I'm an affiliate of Blue Marble Space Institute of Science, and this is an international nonprofit organization here. And of course, Dr. Siddharth Pandey is a member of that organization as well. And I really love working with them because it gives me a platform to communicate and work with people internationally, something that I do work at NASA Jet Propulsion Laboratory, but there are more restrictions associated because it's a government agency. So yes, Blue Marble Space is something that really allows me to reach out on an international platform and work with people abroad and everywhere else with no restrictions. And this photo, this is from one of my expeditions. This is where the lunar landers from the Apollo missions were tested by NASA. So it's more like a state park, but now it's just a national preserve monument. It's a lava field, and you can see that. I'm not sure if it's very clear, but that's my tiny figure is me. I'm standing at the entrance of a lava tube cave, and these lava tubes are actually quite young. If you think about in terms of geology, they are only about 2,000 years old or so, compared to some of the other lava fields, which are thousands and thousands or millions of years old that you go to. So this terrain actually provided NASA a good place to test their landing systems when they were designing the Apollo missions, because the terrain is kind of very similar to some of the areas where they had planned where the moon landing would take place. So yeah, that's I really enjoyed exploring this, and they still go. The rocks are not exactly very similar because it's basaltic rock on Earth, so lava flow rocks or basaltic rocks on Earth are still pretty quite different from what we see on the moon. But it's just that jagged terrain and that sharp-edged rocks is something that is important when engineers are trying to design systems landing on the moon. So how did I become interested in space is an interesting story. And I wanted to start there because I thought it may resonate with some of the students that are here. It started when I was some single digits old. May have been 6, 7, or 8. I don't quite remember that. But I'm not sure if a lot of you have faced this in your personal life. In the summers, many families, and I come from a middle-class family, We did not have air conditioners back then, of course, in my home. And in the summers, it was just easier to sleep on the terrace. And this is common practice in many families across India. In many villages, small towns, small cities, people do sleep on the terrace under the open sky. And so every summer when I was sleeping under that open sky, and of course, the sky at night is beautiful with all the star systems and constellations. So that would keep me awake for long periods of time in the night. And I was kind of a difficult child. I wouldn't go to sleep easily, so I would insist that my dad tell me stories, and it was a pain for him many times, but he would always comply. So yeah, he would sometimes he would make up stories, but then other times he would tell me stories about the Apollo missions, of how there was this place called NASA, and they had landed people on the moon. And because the Apollo missions happened when my dad was pretty young, so he remembered all the excitement around the world, watching everything on TV. So he would tell me stories about that. And then every once in a while, he would just explain the basic astronomy he knew from his education. And he'd tell me that, some of those stars, some of those dots that you're seeing are actually not stars, and they are planets. And some of the dots you're seeing are probably entire galaxies. And that's when I first started to grasp that concept of the difference between these things. What is a galaxy cluster, and what is just a star, or what are planets? So it became very fascinating to me. And of course, as a little kid, like all of us, I think, who are in space, we want to be astronauts. So I indicated that, hey, I want to be an astronaut when I grow up. So my dad thought, of course, she's a kid. So yeah, he was like, sure. But then, of course, when I grew up, that's not a serious career in India. If you tell your parents you want to be an astronaut, I don't know how many times parents are like, yes, go for it. It's got to be an uphill climb, almost equal to climbing K2 maybe, but it's not something that's encouraged. And yeah, my family actually encouraged me to, my parents really wanted me to go into medicine and become a surgeon. But somehow I was never into medicine. I was just not attracted to the field as much as I was into space. So it wasn't until a very long time after, when I was an undergrad and pursuing biotechnology, that I came across astrobiology, and I was like, okay, this is my chance. This is all the knowledge I'm gaining in biology. Maybe I can apply it to what I really love, which is space research. Because to be honest, if I was left to make my own decisions after high school, I would have gone into aerospace engineering or mechanical engineering or some form of engineering. And I'm the only scientist in my family. Everyone else is an engineer of some kind. So yeah, I went into biosciences and learned about astrobiology. I did have to teach myself a lot. And initially, when I indicated to my family I was going to take this up for master's, they were not happy about it. So it was a long battle from there. For my master's, I went to England, to University of Nottingham, and again I was taking up a upper level biotechnology course that lets you work in the pharmaceutical industry or any other biotechnology industry. It was applied biomolecular technology, and after working there, I had an internship at EMBL, which is European Molecular Biolabs in Germany. I was still doing medical science focused biotech research. And then I seriously started to think about doing a PhD, because that's something biologists eventually think about, how to progress in their careers. And I got some really good advice from some of my professors. They said, only pursue a PhD in something that you really, 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 really love, because PhD is very hard, and you will only survive that if you really love what you're doing. Sometimes things will get so harsh that a lot of people drop out of PhD, and that's something that we see quite often in universities in the US, where people join a PhD program and then they never finish because of whatever reason. So that was great advice. And when I really questioned myself, it was always astrobiology that was ringing in my head. And I was like, you know what? I will have to take that leap and transition towards astrobiology research. So I applied to a PhD program in the US and got in. And then, yeah, it was because astrobiology, like your director mentioned, astrobiology is a very vast field. it's usually a coming together of two different fields of sciences. You bring them together and you apply it towards space research. Or you bring together engineering and some science and apply it towards space research. So for me, with my background in biotechnology, which was mostly micromolecular biology, I combined that with a lot of geology and geochemistry towards exploring the project that I did, which was mostly how to look for biosignatures on Mars, something that Perseverance will be doing now when it lands next year. So my first experience working in an astrobiology lab was not in US, it was actually in Germany right before I started my PhD. So this is at the German Aerospace Center in Berlin, and this is the lab where I worked in. So this photo was taken back in 2011. That's when I was there, summer of 2011. They were working on the preliminary experiments, biology experiments for the ExoMars mission, which will now again fly. Previously, when I worked on this project, ExoMars was scheduled to fly in 2018, but they have been pushing back the mission, and now I think it's going up in 2022, if I'm not mistaken, because of various engineering challenges that they face. But yes, back then, the astrobiologist I worked with was designing, doing some experiments as ground control to design the rover experiments that would look for life signatures on Mars. So they had designed this very complex and interesting Mars simulation chamber, which simulates the atmospheric conditions, the gas mixture, and radiation conditions on Mars. And they were testing various organisms that they had collected from around the world. So we had lichens that was collected by my PI from Antarctica, dry valleys, because that's a pretty good analog site of Mars. It's really cold, it's really dry. So they found these lichens that grow in that area. They got those back. They had some cyanobacteria from Atacama Desert and some other organisms that they were testing inside this chamber. So I worked with the lichens. And the experiment that we did that was really interesting and that really got me hooked towards thinking more towards Mars was these simulated lichens under Martian conditions to see if they were photosynthesizing like they would in Antarctica. And at first, when we exposed them to direct Martian radiation conditions, they died. They did not survive direct radiation on Mars, which we know is Mars radiation is pretty high because of the lack of atmosphere there. But then we did something different. We actually simulated, say, if you have these rocks and giant cliffs of Mars, and sometimes those they have crevices or cracks and fractures, and you can have lichens growing in there, or in a more hidden environment like a cave, if you have lichens growing on there. So we kind of simulated that. We put like a kind of a rock that would shield the lichens from Antarctica from direct radiation inside of this chamber, and then tested. I just realized it was not on full screen for some reason. Sorry about that. Okay, so and then tested the photosynthetic activity of the lichens, and we noticed this interesting trend. Let's move this here. Interesting trend that the lichens from Antarctica actually started to adapt to the Martian condition and was photosynthesizing normally. And this small break, so this small break, or this red line, this red box that you're seeing, this here is showing us that there was some kind of a malfunction in the Mars simulation chamber when things were not right. The lichens you can see has gone down. They've gone down in their photosynthetic activity, but then when the mechanical problem was fixed, it actually went back to its photosynthesis, normal photosynthesis. And I saw a quick question there, why not modify the organisms? Yes, modification is a whole different thing I can talk about maybe during the panel discussion. The aim of this experiment was just to see whether Earth organisms can adapt to conditions on Mars. That was the primary aim. So, and Earth organisms that are more extremophilic in nature. So these are lichens not taken from, say, your backyard, but they are taken from Antarctica. So they're coming from an environment that is more Mars-like or a Mars analog site. And we wanted to test whether it would really survive Martian conditions. So that was the goal. We can talk about genetic engineering in a little bit later. So that was cool. And this experiment really got me hooked towards Mars. And I kind of focusing a lot towards Mars research. How are we looking for life? What are we planning to do? We send humans there. And something that has always intrigued me or piqued my interest is being able to grow crops on Mars. Even though that was not my primary PhD project, I've always done it as a side project or kind of a side hustle. I've always had that. So at Washington State University, where I was doing my PhD, this is more towards the end of my PhD when I got assigned as a payload advisor for their students engineering group who had, so NASA does this thing at universities in the US where you can win competitions and fly your payload with NASA, something that in India also does, where universities like Amity is going to be flying experiments with ISRO. So the team at Washington State University was this large team of engineers that had won a competition to fly a CubeSat. So CubeSat would just be 10 centimeter by 10 centimeter. And they approached me as the astrobiologist on campus that, hey, if we want to send a payload and you want to test something, what would you like to see? So of all things, at that time, this is the year 2018, I was thinking very heavily about the future of crops in space. And my thought process was that, at all times, there may not be astronauts or there may not be people to actually interact with the plants or when the crops are growing. So if we are setting up structures, whether it's in the lower Earth orbit or if it's on the moon or Mars, at some level or at all levels, it would have to be automated. So that was my thought process. And I was like, okay, if you want to automate things or if you want to build automated structures for growing crops, what would be the first step that you would test? And yes, the way we grow plants, say on a extraterrestrial body, say, for example, Moon and Mars, it may be more hydroponically associated than how it's grown currently on the ISS. Or say if we put in an entire structure in the lower Earth orbit of just dedicated to growing plants, then it may not be hydroponic completely because water behaves very differently in space. So it's more substrate based. But then I was like, I told the students, let's test a hydroponic system in low Earth orbit because I'm pretty sure nobody has tested that. So we came up with this seed chamber, and this is just a CAD model. Just know that the measurements are really small because in a 10 centimeter by 10 centimeter CubeSat, the plant chamber itself is extremely small. We had a little bit of space assigned inside of that CubeSat. And yeah, this first picture, you're actually just seeing our ground control experiments where we were testing our carbon dioxide sensors in a kind of a bioreactor setup that I had made. So I just had green pea seeds in there in a little bit of water. And I wanted to test what happens, what are the changes in carbon dioxide when germination takes place. So that's the only reading we were trying to take. But then in the actual chamber, once this CubeSat is released into the lower Earth orbit, there will be some water that will be released into the seed chambers here. And then we are just going to wait to see if they germinate in that environment, in that we have some amount of thermal activity going on because of the systems itself, because of the Cube satellite system itself. But mostly it's going to be a much lower temperature. So we have tested we did control test some temperatures, lower temperature germination, and we were able to see some good results. So we are going ahead with that. And we have about 8 or 10 green pea seeds, not a lot. And once that's released, the carbon dioxide sensor will take a reading and stream it back to the data control center at Washington State, telling us there was a change in carbon dioxide. And we have also put a really tiny camera in there just to see if it can also send us a picture. We are not highly optimistic about the photos of how, in a very tiny CubeSat, if we'll be able to get clear photos, but we have done that with some LED lights that are on there that will be turned on. So we'll see. This is scheduled to fly. It was actually supposed to fly this July, but because of COVID-19, the flight has now been pushed to January. 2021. So this goes up to the International Space Station, and then they release it to the low Earth orbit. So next year, sometime, we'll find out if the autonomous germination happened. So autonomous greenhouses is something that I'm thinking about, and this is just a first step, really amateurish experiment that we are trying to do in the lower Earth orbit. But otherwise, on my full-time job right now is studying spacecraft. Let me move this window aside. Yes. So studying spacecraft microbiome for planetary protection and sample return. And again, your director said that when you're sending live organisms to the moon, there's a question of ethics. There's a question of ethics, and there's a question of international policy. So planetary protection is this agreement between or among spacefaring nations in the UN, where they have agreed to comply with the regulations of not forward contaminating another terrestrial body when they explore it with rover missions or experimentation. And all spacefaring nations have signed that treaty where they have said that, okay, they will not release any Earth organisms out in the open environment of, say, the moon or Mars or any other planetary body that we explore. So this is why NASA or missions at JPL or the rovers at JPL that are built, or any spacecraft that are built here undergoes a very extensive process of sterilization. And we are always forever evolving the methods in which we sterilize our spacecraft instruments without damaging them, of course. So one of the things that we have done, or one of the things that NASA has done, this was even from before I was born, when they were sending rovers to Mars, is they have collected, after the sterilization process is over, they actually take swabs and take wipes and take samples of anything that may be on the spacecraft, and then culture those microorganisms or store to create an archive of storing those microorganisms in -80 freezers and then characterizing them so that we know we have data on whatever may have hitched a ride on one of these rovers to outer space or to another planet. So currently my project is actually characterizing this archive we have for all the Mars rovers since the 1970s. So this is starting from Viking and working my way all the way up to Perseverance. So mostly the bacteria that we see are spore formers. Those are the ones that usually survive the heavy chemical treatment or heavy radiation tests and a heavy radiation treatment that the spacecraft undergoes before launch. But yeah, we definitely document those, and it was especially more important. So the planetary protection exercise was very important for Perseverance that just launched, because Perseverance is the first rover that will actually take soil samples on Mars to be returned in a future mission in which we look for life. And we had to make sure that it absolutely had no spores on any of its drill bits. That was probably the hardest parts that the engineers had to go through. And I was looking at the numbers. It seems planetary protection ended up taking, I think, close to 14,000 swabs from Perseverance rover by the time it launched. So we have that, and now we are in the process of documenting every microbe or characterizing every microbe that may have been isolated from the spacecraft assembly. So with that, I will conclude my talk and pass on to the next panel member.
Thanks, Afshin. Thank you very much.
Yeah.
May I request Ravi, Dr. Ravi Kumar Hosumani, if you could speak. And just in the interest of time, we'll all try to fit our explanations within about 10 minutes. And then we can, if something is left out, we can take that up in the panel as well, because I know it's challenging to talk about so many things. So maybe 10 minutes each person. So over to you, Ravi.
Yeah, sure. Can you please put up my slides?
Just a second, sir. I'll do that.
Sure. So just to give a brief intro about myself, I'm Ravi Kumar Hosumani, currently working at University of Agricultural Science, Dharwad, in Department of Biotechnology. And before I joined IIST Dharwad, so I was working at NASA Ames Research Center for close to 7 years as a NASA postdoctoral program fellow, as well as research faculty there. I spent quite a bit of time with Dr. Shemla Bhattacharya's lab. And in fact, Siddharth was mentioning about that, right? So we were for a couple of years. We were really lab mates there. So I'm going to talk about mainly the fruit flies. And before that, let me thank the organizers for giving me this opportunity. And I would very quickly go through, and maybe we can discuss in detail in the panel discussion, I guess. And also feel free to send me an email in case you guys have any questions on this one, actually. So so basically what I'm going to talk about is my experience, which I have done research on fruit flies and how relevant that model for the understanding the astronauts' health in space. So I'm going to talk about that a little bit on that and what kind of experiments we have done on the International Space Station and all that. Okay, so next slide, please. So basically, you know that I think it is important to, the sort of questions I'm looking at the chat box is, it appears that a lot of people have no idea.
Ravi, I think you went mute.
Am I?
Now you're fine. Now we can get you.
Okay, yeah. So, so I think it is important to highlight why we need to do this spaceflight research on the International Space Station or in space in general, because I think the whole idea is why the people like Jeff Bezos and Elon Musk are spending too much money on that. Is they want, the ultimate goal is they wanted to make this humanity or humans multiplanetary species. It might be down the lane 100 years, 200 years, but before that, before we do that, we need to understand very comprehensively what is the ill effects of space and space flight on the human health. So that's the reason why many people do this kind of research, and especially in the Western world, and many of these space agencies like NASA, ESA, European Space Agency, JAXA, and spending too much money on the space biology. But I mean, of course, it is not at very, it's very initial stage in India, and hopefully they will pick it up. So it is important to understand what are the ill effects of the space flight on the astronaut's health, right, in general. For example, there is huge issues of cosmic radiation. Cosmic radiations can really penetrate your cell molecules, that can damage your DNA. That DNA damage can lead to the kind of cancer-like diseases, which is not very pleasant to look into that, right? The next, please keep clicking on the slides. So bone and muscle loss is another issue we have in the space flight conditions. According to the NASA's data, if you're on the International Space Station, you're probably going to lose 1 to 2% of bone mass and 20-25% of muscle loss, which is huge actually, right? So you need to prevent that. And there is, and good news from that is basically you can reduce this impact by doing the exercise. That's why you can see on the International Space Station, astronauts spending at least 2 to 2.5 hours in physical exercise so that they can keep this bone as well as muscle health intact. And then we have the cardiovascular issues, right? In the space, heart doesn't have to pump against the gravity. As a result of that, you can see the impaired cardiac function as well as changes in the structure as well as muscles in the heart structure. So that needs to be addressed before we even think of colonizing any planets in the solar system. And another issue we have is compromised immune system. In the sense, we know that from the literature that the astronauts came back from the International Space Station as well as from the Apollo missions were highly vulnerable for various kinds of diseases because they were having this compromised immune response. And as a result of that, they were not able to fight against the fungal infection. I'm going to talk about a little bit more on the cardiovascular as well as immune response in my next slides, because we have done quite a bit work on the fruit flies. Then more recently, we're also having some issues with the astronauts' macular degeneration. In the sense, what will happen is in the space, when you are in space, basically the body fluid will flush to your head region. As a result of that, it will put a lot of pressure on your back of the eyeball, which will lead to the either partial blindness or fully blindness. So that these are some of the major health issues we need to be addressing before we even think of colonizing solar system or colonizing one of the planets in the solar system. And of course, our best bet is Mars. And other than that, we also have a lot of psychological stress because the astronauts are staying in confined, isolated area. So that will lead to the psychological stress with the physical damage is deadly combination. So we need to understand the holistic health issues in order to go for the space exploration missions, right? So of course, that's where these model arguments comes into the picture. And of course, if you look at the literature, so far only 559 people have gone to the space. And among them, only 8 people have completed around 300 days missions. So this is a very statistically very skewable data, which you cannot depend on the, working with the astronauts themselves directly. samples, urine samples, and fecal samples, which might be helpful or may not be helpful for extrapolating that biological data to the larger population. I think these model organisms play a very critical role in understanding comprehensively health issues. And why fruit flies? The reason why the fruit flies is, of course, there is a lot of commonality. Although these fruit flies and humans diverged around 634 million or 650 million years back, but you still have a lot of commonality in terms of functions and in terms of genes, right? For example, there's a website called Homophila, where individual genes of the Homo sapiens have been compared with the Drosophila melanogaster, and they come to the conclusion that around 60% of the genes between humans and Drosophila are very comparable. And especially the genes which are causing diseases have 77% similarity. That will make all the more important why these Drosophila tiny model organisms are very helpful in understanding the human diseases and specifically astronauts' health. And of course, they are very tiny. You can make these multi-generation studies because the life cycle is only 10 days. So, easily you can do the multi-generations. Those are some of the advantages we have with using the fruit flies as a model organisms. So if you really look at the history of animals in space, all kinds of animals have been flown, 1952 up until 2020. You take microbes, nematodes, fruit flies, and all kinds of mice, rodents, monkeys, animals, everything has been flown. But really, the next slide, please. Just click on the slide. So really, if you look at before the Laika dog became a, can you go back to the previous slide, please? So before this Laika, the dog became a really celebrity dog, and exactly 10 years before that, in 1947, February 20th, the Drosophila were the first animal to went to the space actually. So it crossed 108 kilometers of altitude. According to NASA's definition, if you want to be called astronauts or you are in space, the definition is you have to go at least 50 miles away from the Earth's surface, right? So in technically, the Drosophila flew to the space in 1947, exactly 10 years before the Laika became the celebrity dog. Next slide, please. So next slide. So from 1950 to 2000, a lot of work has been done on the fundamental biologies, and basically from 2000 onwards, people started looking at the how we can utilize this fruit flies as a model system for understanding the astronauts' health in specifically. And Dr. Shyamala Bhattacharya's lab at NASA AIIMS has contributed immensely to this area of research by sending various fruit fly experiments like FIT, Heartfly, FX, FFL1, 2, and 3 series of experiments. I was very lucky to be part of this team, which flew many of the experiments between 2011 to 2017. Next slide, please. So very first experiment Dr. Bhattacharya's group has done is a FIT, which is fungal pathogenesis, immunity, and tumorigenesis. Basically, you can see this is done in the STS-121. So we have these tiny cassettes, we call it as fly cassettes, and these have around 50 to 100 flies. Once they exposed to the International Space Station, they receive these flies back, and they looked at the gene expression studies that clearly shows that there's a significant downregulation of defense-associated or the antimicrobial genes, which are very critical for the innate immune response of the flies, were significantly downregulated compared to the ground control. Next slide, please. Keep clicking on the slides. Because of the time constraint, and I want to just go with quickly, and we can spend much time on the discussion. So again, this particular fly is very interesting. Actually, taken by one of our collaborators from the UC Davis. You can see that this particular fly born and brought up in the space or International Space Station. As soon as they received these flies, they injected a very small amount of Bavaria bassiana fungus, which clearly, within a few days, it spread all over its body. You can see the white patches all around that, which clearly shows that these flies came back to the International Space Station were highly compromised in terms of their immune response to fight against this fungus. So how relevant this information to the human specimens or astronauts' health? I found this interesting paper actually because this has been published in Brain Behavior and Immunity in 2014 paper. I would like to grab your attention to the C panel actually. This is a phagocytosis. This is the macrophages which are equivalent to the plasmadocytes of the Drosophila, which clearly shows that again the phagocytosizing ability of these cells were significantly downregulated, indicating that there's a compromised immune response in astronauts as well, which is exactly what we have seen in the Drosophila as well. So again, there are much more publications on that. So that clearly shows that flies can be a very good model in understanding the innate immune response in space, and which is very relevant to the what we have observed in the humans or in astronauts. So the next experiments carried out when I was in NASA Ames Research Center was hard flies. Of course, this is the whole main objective of this experiment was we wanted to understand the impact of space on the heart structure and function. And of course, it is not as well defined as the human heart. It has a very just simple tubular peristaltic pump. But it is, if you look at the genetic and molecular function of cardiogenesis and cardiac function, there is a significant overlap between that, and it is very important to understand the heart flies in the fruit flies, which is very, very relevant to the human hearts. I'm going to go to the next slide. So I think this is what I wanted to highlight, because this clearly shows how cardiac function is very relevant in humans and the fruit flies, right? There's one publication, Wolf 1991, which clearly shows that as we age, humans from 50 to 90 years old, there's a clear indication that there's an arrhythmic movement of heart happens, right? Similar phenotype also we have seen in the fruit flies. 1 week old, which is equivalent to the 10-year-old boy or a girl, you can see that how consistently this heart is beating. Whereas 7-week-old flies, which is 70-year-old lady or a gentleman, which clearly shows that there is arrhythmic or a very inconsistent beating of this heart, which clearly shows that this phenotype is very, very relevant to the human heart. Again, this has been done by Dr. Bodmer et al. At Stanford Burnham Institute in La Jolla. So what they have done is basically they took these flies and sent into the space and bring them back and look at the heart of those flies, and they have seen there is a significant arrhythmic movement in the heart. So again, how relevant this one with the astronauts' health? There is one report which clearly shows that the ventricular tachycardia is nothing but, can you please go back to the previous slide? That ventricular tachycardia is nothing but impaired cardiac function is very relevant to what we have seen in the Drosophila melanogaster. And of course, you can also see the cardiovascular issues, the deaths in the Apollo astronauts, which shows that cardiovascular issues are very, very important in order to understand their the impact of spaceflight on the cardiovascular health. And you can see this is a blue bar, which shows that there's an increase in the deaths in the astronauts which went to the moon mission. So it's, again, this is very relevant model in terms of Drosophila melanogaster. So next slide. So I'm not going to talk about much about this one because already Dr. Siddharth has covered, and in fact he was one of the member when we flew this experiment on the Space Explorer mission. Just wanted to understand in the physiological stress and the behavior in the fruit flies, because it's very relevant in terms of human astronauts in the space station, because we know that the astronauts which are staying in the confined area have already elevated level of physiological stress that could impact their behavior, right? We wanted to understand that in this spaceflight experiment. Next slide, please. Next slide. Okay, so with that kind of experience, once I came back to India, and we were, I was very keen on, initiating this kind of research in India. So then we, luckily, coincidentally, there was an announcement by the government of India, Gaganyaan program, and ISRO announced for the opportunities. We grabbed this opportunity, and we, in collaboration with Dr. Jija Lakshmi, IIST Trivandrum, we proposed this study on the kidney stone formation in fruit flies. Again, this is very relevant for the astronauts, because you know that because of the bone leaching and bone minerals will get deposited in the human kidney, in the ISS, or in the space in general, as a result of the kidney stone formation. And similar phenotype also you can see in the Drosophila as well. Drosophila malphizian tubules are equivalent to the human kidneys. So you can see that beautiful picture where calcium oxalate is deposited in the malphizian tubules, similar to what we have seen in the kidneys in the humans. So we wanted to see how exactly this kidney stone formation differs in space and on the ground. So hopefully, this has been recommended by the ISRO, and we're finger crossed, and hopefully it will fly soon. Next slide, please. Okay, I think I just rushed through because I think we can spend too much time on the panel discussion rather than giving all these details of experiments I have done. So the overall is what I would like to conclude here is basically fruit flies, although they diverged from 650 million years back from the humans, there's much functions, and as well as in terms of genetics has been conserved between those two, and it has like 6 Nobel Prize in physiology and medicine. So that clearly sums up how relevant this model for understanding the human health, specifically astronauts' health. And Dr. Sharmila Bhattacharya's group in exploring this kind of research using this tiny fruit fly model. And I would like to thank all my lab mates and Sharmila sitting there for doing such kind of work and inspiring people like us to do.
All right.
Thanks a lot, Ravi.
Thank you.
Thank you, Ravi. We move on to Dr. Luis Zia, who is joining us from Boulder, Colorado, US. Luis, take it away.
Good morning. Good evening. I think there is some mic. Sure you can hear well. All right, I'm going to try to share my screen. All right, can you see my slides?
Yes.
Perfect. All right, I'll try to stay within the 10 minutes, but if I don't, start waving at me. Otherwise, I'll just keep going. Thank you very much for this invitation. I'm honored. As Siddharth Pandey, Dr. Pandey knows, I'm pretty excited about international collaboration for space exploration and space science. So I never had a chance to reach out to scientists and students in India, so I'm pretty excited about this opportunity. Thank you. My name is Luis Zia. I work at Bioserve Space Technologies at the University of Colorado Boulder. And we send biological experiments to space station. Either we help scientists translate their experiments so that it can be implemented on orbit, or I serve as PI from the science side for our own experiments. So I've had the opportunity of working on 18 flight experiments, which is really cool. Every time you see one launching, something gets in your eye and then the other eye. And today, I think what I'm going to tell you is very complementary to Dr. Hossamani's talk, because I'll say a few of the same things, but from a different perspective of why do we do these kind of experiments in space? And at least the focus I try to do my research is the use of the phenomena that we see in space to help novel solutions to medical problems on Earth. So why would we even do it, right? So I'll do a very quick time travel. I'll go through 4,500 million years of history in about 45 seconds. And for that, I'll take 3 pictures of Earth. The first one is on the Archean Eon, about 3,500 million years ago. And then I chose one parameter that is relevant to life as we know it. And here is atmospheric molecular oxygen. So you can see there was a very, very small amount of that in the gas on planet Earth back then. And the reason for which I chose this period of time is because this is where we see the oldest fossils. At least that there's no, that people agree that these were live organisms. Next, we're just going to go 500 million years ago to the Cambrian period. And the atmospheric molecular oxygen has increased to about 12%. I chose this one is because this is where we see the biggest diversification in history of life, to the point where 90% of the animal phyla that currently exists was emerging at this point. And finally, today, as most of you know, we have about 21% molecular oxygen in the atmosphere. And when you see this as a clock where we start at noon, the first laugh happens between 2 and 3 in the morning. And then the Cambrian explosion that I was referring to, it's around 10, 11. And then on the last second of the last minute of the last hour, we show up. So there's a lot of things that have changed. I spoke of one parameter, molecular oxygen. But think of it. The presence of oceans, the salinity, the temperature, composition of the gas, composition of what's in the water, all of these things have changed. And for 3,500 million years, life has evolved adapting to these changes, but something has been constant, and that's gravity. And now that we have the ability to bring cells and organisms to space, we can treat gravity as an independent variable. And that has allowed us to see phenomena that otherwise is not observable on Earth. And with that background, I'll give you a few examples of the kind of microgravity research that we've done and supported where I work at Bioserve. One example is cancer research. And what you're seeing is a picture I took on the microscope. This is human adenocarcinoma from a 26-year-old male. Those are the clear little bubbles that you see there. And we've flown several cancer research-oriented payloads to station. What you're seeing, there's astronaut Peggy Whitson holding one of our biocells. That's called a 6-well biocell. We have incubators, refrigerators, a microscope, cameras. We have a lot of hardware on station since 2001. And then we did serve scientists to implement their research in space. So in this case, the reason for which we were doing cancer research on orbit is because you can have these cells form three-dimensional structures that look closer to what actually happens in the human body. So when Peggy Whitson was doing these experiments on orbit, we would see live in our payload operation control center how the cells looked. And she was traveling on the z-axis, and you can see how she goes forward and backwards and still see cells on focus. After that, I will run downstairs to my lab and run the ground controls. And as soon as you change a little bit the nub, you lose everything of focus, because on Earth, they tend to be flat. As soon as another one gets on top and another one on top, gravity just pulls them down. They're large enough to be affected in that fashion. So when you have something that looks closer to what actually happens in the human body, you can have less false negatives or false positives when you're trying to test a molecule to see whether you can make it through the pipeline and make it a drug available to people. Another case, and this is something that Dr. Hasamani talked about, is osteoporosis-related research. If you're sitting, you may be thinking that you're not really doing anything, but in reality, the weight of your body, and your weight is a function of the mass and the gravity, right? 9.81 meters second square on Earth, on the Earth's surface. that weight puts a stress on your bones and your muscles, and it is your skeletal muscles that keep you upright, and it is those bones that are being compressed that are maintaining that structural stability of your body. So what happens when you go to space, right? So you don't have that stress. Homeostasis, the body understands like, hey, why would I be wasting all this energy maintaining bones and muscles that I'm not using? So they start reabsorbing them. In fact, on Earth, the worst type of postmenopausal osteoporosis, if it were to go untreated, translates to about 1.5% bone mass loss per year. But on space, if you don't do anything, it's higher than that per month. So this is a problem for human space exploration, but it's also an opportunity for us to utilize this environment to test molecules to see if they can work against osteoporosis. And we've done this in the past. We've supported payloads from pharmaceutical companies and NASA. Actually, this experiment here is a multiple party with NASA Ames and Amgen, and translated into a drug being put on the market in 2010. For perspective, in 2012, it sold $1,200 million. So there is that side of why we do this as well, is the commercialization of this. And the aspect of what I was saying is that this can also help people on Earth. And then muscle is something similar. What you're seeing here, these are not animals that do steroids or go to the gym 2 times a day, 7 days a week. They have a mutation on the myostatin gene. So they do not synthesize a protein called myostatin that tells the body to stop building muscle. And that's another experiment where we supported flying cohorts of mice. Half of them would get a molecule to see if they could inhibit the myostatin process, so that people who are bedridden from different diseases, cancer, AIDS, a bunch of different diseases that make people be on bed for long periods of time, and therefore have muscle reabsorption, to see if this can be translated into a drug for them. In a different fashion, this is a sperm experiment we flew recently. For the PI is Dr. Joe Tash from University of Kansas, and we flew human and bull sperm. No, we were not trying to make a minotaur. This was a study to see if when we go to space for very long periods of time, humans will indeed be able to be a multi-generational species. Are we gonna, the whole process, is that gonna be even possible? And the first point here was to characterize the motility and just protein expression and different parameters, basically, of the human sperm. And there were no eggs present. This was specifically for the sperm. Probably one of the coolest things is that this is one of the ones that got a lot of attention, especially from different outlets. This is the Daily Show with Trevor Noah. He had a whole sketch on it. Actually, my hands holding a syringe with sperm made it to Trevor Noah. I should probably put that on my CV. And one of the coolest things of this is the patch. The principal investigator, his brother-in-law is Matt Groening, and he's the creator of The Simpsons. So he got Fox to make the patch of. You see Homer Simpson riding a bull sperm, the space station in the background. So that's pretty sweet. I just gave you a few examples here. We went from a human adenocarcinoma, we went from cancer, fertility, you see osteoblasts. We also do C. elegans. Really what I focus on is bacteria. And there's a lot of reasons for that. One is that for several experiments, we've seen that you need higher concentrations of antibiotics to inhibit bacterial growth. Again, a problem for human space exploration, but also an opportunity to find different channels, things that we don't see on Earth. And in fact, we've seen a suicide gene, an apoptosis gene, overexpressed 24-fold, and the antitoxin underexpressed 40-fold in space with respect to ground controls systematically under a specific condition. So that's also an opportunity to see if if that suicide gene, we can make bacteria kill itself basically when we cannot anymore with currently existing antibiotics. So I'm going to stop it there for the sake of time, and we can leave some time for chatting. Yeah, thank you.
Thank you very much, Luis. That was amazing. And we'll move on to our last speaker, Dr. Sangram.
Hello, can you hear me?
Yes, yes, we can hear you. And we can also see your slides.
And thank you very much to the organizer for giving me this opportunity. And it's really an honor. It's an exciting panelist and exciting students. I can see there's a lot of questions in the chat session. So definitely I'm not a space biologist or astrobiologist. Basically, I'm a synthetic biologist. So I mean, many students ask and also I've seen actually told that what could be the background. So my background is I have a double bachelor's in chemistry and BSc in chemistry and BTech in chemical technology. And then I moved to University of Toronto and I did my PhD in a chemistry department, but in the synthetic biology. And then again, I moved for my postdoc to Amity in a chemical engineering department, working on the systems biology and some other technology. And then I came back and joined 6 years back at Saha Institute of Nuclear Physics in Kolkata. So what I'd like to see that what the space space travel, how it is going in the future. So if you see, this is a poster from the NASA. And that's, they said, what is the potential of the synthetic biology in the space? Now, today's Mars mission could be definitely different. No, please go to the previous slide. Please go to the previous slide. Yeah. So, so the this one. So the whole idea of the plan, so if you go to the Mars, it is not like just going to the moon right now, because that time in 1960s, 70s, people, I mean, human has to show that they can go to the moon or something. At this moment, if you want to go to the Mars, you have to go for, it will take like a year or 2 years. And if you want to come back, it will take the same amount of the years. So the whole idea, if you want to go to the Mars, the first thing is that not to colonize Mars, but to develop a research base. If you have seen the movie Martian, and if you've seen that, they have a research base. Now the question is, if you want to send a bunch of astronauts to the Mars, so there's a huge amount of resources you have to take, because it's a question of years, right? So if you want to have some food like burger or some dosa, you cannot have it, you cannot take it from the Earth, right? So you have to make it on the Mars, not just the cooking, but all the ingredients you have to actually develop in the Mars using whatever the resources the Mars has. So this is a big challenge to going for the different extraterrestrial bodies. So what NASA thinks, or many people think, that synthetic biology would be the key. Why? Because you don't need to everything for your space mission. Just pack synthetically engineered organism, okay, and in few vials, and go to the space, and those organisms will do everything for you, starting from making the roads, making your energy, making your materials, the drugs, foods, radiation resistance habitants, whatever. Just those engineered bacteria or any other organism can do that. So that is the whole idea, that could be a solution if we can do that for this kind of space mission. Could you go to the slide, please? Next slide, please. So in 2015, actually, Adam Erkin's group, actually, they wrote a proposal paper, which it's like this slide. So they said, what are be the grand challenges in the space synthetic biology, which could actually change the human travel? And they identified 6 areas, which includes the resource utilization, the manufacturing, life support, space medicine, space cybernetics, and definitely terraforming. I would say para-terraforming other than the terraforming. So these are 6 areas where the synthetic biology can actually make some difference. And could you go to the next slide, please? And now I'd like to discuss that what is the special of the synthetic biology which can actually make this difference. Next slide, please. Previous slide, sorry, previous slide. Slide number 3. So what is the synthetic biology and the systems biology? It's a conceptual shift in the bioengineering, which started from the end of the last century. Before human, I mean, in the last century, people used to think that any biological function depends on a single gene or protein. That's why people try to look for a single, for example, a single marker for the cancer, but they never found it, right? So the whole idea of the engineering project was that if you actually find all the things of the human body, then you can actually treat any disease, including the cancer. But that didn't happen. The reason it didn't happen, not understand the function on a single gene, but the interactions of all these genes together. And now this network is very complex. It is so complex. There is no mathematical, there's no mathematics to actually decipher the whole complexity of those kind of the network. However, from the last century, there is some of biological network with the electrical engineering network. So one way to understand that biological networks could be from the knowledge of the electrical engineering network. So this is a complementary approach. Definitely that is not the final, but you can do some, you can understand something better than what is conventional at this moment. That's a complementary understanding. So if this philosophy is correct, then it is also possible that we can actually develop some of the genetic following those electrical engineering people and put inside the cell, so we can develop a new program or a new function, or you can actually do the new programming. Yeah, next slide, please. Is it slide number 4? Because somehow I cannot see that. Is slide number 4?
Yes, sir, this is slide number 4.
Okay, I couldn't see. Anyway, that's fine. So basically, what the lab is mainly interested synthetic biology, in terms of synthetic biology, is that if we make a bacteria artificially intelligent, whatever that means. So we would like to make the bacteria as an artificial neuron in terms of definitely not, I mean, simpler, most simpler. It's not a neuron, but an artificial neuron, which is a generally a mathematical term for the artificial neural network. So next slide, please. So here is just an example where I can tell you that if you see what we try to develop, something called a 2 to 4 decoder. And this decoder is a part of the communication system.
Dr. Sangram, I think we lost your audio. Are you with us?
Sangram sir, you can try switching off your video once, maybe that would save some bandwidth so that the presentation and audio can continue uninterrupted.
Can you hear us, Dr. Sangram? Okay. Is Dr. Sangram with us? Can admin please confirm?
He is visible. I suggest, shall we wait for a minute or so?
Yeah, let's wait a minute. It's 8:59. We're a bit behind
Looks
time.
like Dr. Sangram has joined through 2 accounts right now.
Okay.
Can you hear me?
Yes,
Yes,
we can.
So here is the decoder. What you can see in the left hand side in the top. So that's an electronic design of the decoder. And what we developed a bacterial neural network for the decoder, which is the bottom panel of the left hand side. And then so we do all this kind of engineering on the cell, the mathematics. And then when actually do the experiment, you can see in the right hand side, this is a is the same as the truth table on the top left. So this way I can say that synthetic biology can do something in different way, design-based biology, not general biology. So here I want to go to the human space exploration to the Mars. They actually go through the microgravity. And this microgravity is like this. And definitely microgravity is basically a health hazard. That's all the speakers actually show. And way, so one way it can affect the health is the deregulation of the immune system. And second, and the other way, there is a
So we lost you again.
Dr. Sangram, I am not able to hear you. Your audio is not coming through. Hello?
So the current statistics cannot actually work on that. So what do we develop? Use all the data on the space data, and then our assistant biology pipeline, which comprise different kind of statistics as well as some machine learning algorithm, actually gave a lot of different insights from that. So we showed that there's a increased risk of cancer. There's a lot of unexplained facts to explain. So if you want to, we can actually go to the publication on that. Next slide, please. So we have also done
Dr. Sangram, your audio is not coming through, but we are having a look at your slide. Dr. Sangram, can you hear us? Okay. Dr. Sangram, may try unmuting yourself. Okay. I'm just a bit conscious of the time. We are about 15 minutes over. Dr. Sangram, if you can hear us, if it's okay with you, we have received, I think, probably most of your presentation. Can you hear us?
yes.
Hi, sir. Your audio is breaking. Can you try speaking something?
I don't know, still.
Sangram sir, I think it is clear now. Can you try once with switching off your video and keeping just the audio on? That should help.
Hello, can you hear me now?
Yeah, loud and clear, sir. Please go
Okay,
ahead.
sir. So okay. So basically, I think you were in the slides where I showed like a synthetic small regulatory RNA circuit, like, right? The first biochemical device was sensing microgravity. Yeah, if you are, so what we actually developed, we developed a synthetic gene circuit, which you can see, and we actually run that through a basically microgravity simulator. And what you can see on the right-hand side, so in terms of GP expression, it gives around 30 times difference between the microgravity. So one of the advantage, next slide, please. For this design is that you can actually target any gene in the bacteria, in fact in the E. coli, and you can actually control the gene expression in the microgravity for any gene. So what here in the next slide, what you can see that we target the cell division process. So develop the synthetic gene circuit, and basically in the result you can see in the right hand side there is some elongated cell in the Earth's gravity, because we are directing the FTSG protein of the cell division protein. Now, if you take that those cells in the microgravity, you can see that the cells have got rescued. And this way, we can actually target so this way we can actually target any cellular processes through the microgravity. And next slide, please. And so just simply, I'd like to tell that we have the synthetic biology could be the key for the future space travel, and our systems biology pipeline get more molecular insight for the microgravity, and our synthetic biology work shows for the first time to integrate microgravity as a physical signal with biochemical process in a human designed way. Next slide, please. And what our experiment has been definitely got through the, got a green note from the ISRO to send you the Gaganyaan mission. What we're trying to do there, we chose a set of the bacteria which causes diarrhea. They're high balance in India, not in the Western world, and you can find in every healthy Indians, and not tested in the microgravity, because those bacteria is not in the Western world, so the Western astronauts not going to have an effect on that, but Indian astronauts may have an effect on that. And basically what we're sending those bacteria to the Gaganyaan mission and try to understand what is happening. And next slide, please. And that's my last slide. That's just, I thank you, all of you, and my lab mates, my students, so my lab mates, and at the very same time, my funding body. And thank you for your attention.
Thank you very much, Dr. Sangram. I'm very delighted to hear about this excellent work that is taking place. I was not aware of the work you've done, so I'll be getting in touch with you and learning more from you. So with that, I think we will now get into the panel discussion, and I'm very, very happy to see that a lot of students are with us, and I will try to ensure that we finish by 9:30 and we don't go beyond that, 9:30 p.m. India time. So let's do a panel discussion for about 10 minutes, and then right after that, we'll go into Q&A, because that's really important for us to take the questions which the students have asked. So to start things, what I can perhaps do is pose a question. I have 3 questions with me that I had written down, and I can pose the question to the panelists. And if the panelists can maybe answer the question in a minute, or maybe say 2 minutes each, then we can perhaps try to go through at least 3 of these questions. It's going to be challenging, but feel free to interject and speak as well if you would like to. And the others can perhaps keep their mics on mute. So I think the first question that I have for everyone here on our panel is all of us are perhaps involved in our different capacities on space projects. Given the fact that the International Space Station is, of course, one orbiting space platform, and India has plans to have the Gaganyaan mission and then move on to having another space station, an Indian space station in the future. What are some of the challenges in your mind that we as a community here on Earth, on ground, will be facing in terms of space biology? What are some of the key things that come to your mind that in the next 10 years are going to be challenging for us? So maybe Dr. Afshin, in the same order, maybe we can just have some thoughts, maybe a couple of minutes from each of you.
So I'm putting myself on a timer to answer that. I think the biggest challenge that we face even today and in the past is bringing countries together, because something someone said here that because space, when we explore space, we should explore ourselves as a species, as the humanity. But that is something that we don't see immediately, and that's why China is thinking of building the space station, and then India has to think about building their own space station. Why can't we all be the part of the same international space station? I thought that's what international is supposed to mean. So yeah, getting through the politics of that and getting people to work together, I think we have so many brilliant questions just coming from undergraduate students, and then all this amazing research that's done by Indian scientists. We can see the potential of, if we join forces of countries coming together, we can answer much more challenging questions rather than doing just basic research. So I'll stop here.
Dr. Ravi?
Yeah. So I think the biggest problem I think I would say is basically so far in terms of exploration point of view, we have looked at only around 340 days time point, right? The recent experiment done by Scott Kelly and Mark Kelly, right? So that is only for a year experiment they have done. If you're really exploring, if the next 10 years, if you're really planning to go and explore Mars and other planets, you need to have a comprehensive understanding of health issues that needs to be addressed, and which is not very it is not very coordinated in the sense in the various space agencies. For example, take in ISRO is nowhere in a position to do any kind of space biology as of now, and so that will be a problematic in terms of space exploration point of view.
Hi, Luis.
Thanks. That's two main challenges. One is to make sure we can keep humans safe during long-term missions. And the other one is something that was spoken about, which is the utilization of biological systems for the benefit and for the sustainability of these explorations. So meaning from the first side is making sure that you can make a list of what are the worst things that happen in terms of detriment to human health as they are exposed to both microgravity and deep space radiation, which are the two hallmark space travel and environmental facts that we need to keep in mind if you're inside a spacecraft. And several of them were mentioned, right? Bone and muscle reabsorption, cardiac and muscle and eye, and there was a list of those, right? So I think making sure that those are addressed would be the first of the two that I have in mind. And the second one is how can we use these organisms to help us achieve what we're trying to do on an overall space architecture. And several of them were commented, including the use of organisms to help us mine. And that's something that we do. The research on is space biomining. We're using Shewanella onadensis to extract iron from lunar, Martian, and asteroid-regolith simulant, so that afterwards it can be easier to segregate, and then you can feed it to 3D printers, for example. And then you can build things. So I think those are the two main buckets under which I would put the challenges ahead.
Dr. Sangram, your thoughts?
Yeah, I'd like to, all the questions are answered. So I just like to take the challenges from the student perspective, because a lot of students are asking that if I am a chemistry background, I have this and how can go. So this is another challenge. So what is that? So this is some mental barrier you are forming within yourself. I'm just talking to the students actually. So that it doesn't matter what is your background. Any science background, I mean, you can see, like, as Professor Pandey actually say, he has some different background. Director at IIST has a very different background. My own PhD supervisor, David Macmillan, so he was a space engineer, and his work was on the, basically his PhD research actually gave the idea that how the robotic communication in the rover's transmission happened. But he, in postdoc, did the synthetic biology. He joined the Investorant in the department of the chemistry. So it doesn't matter what is your background, because when you are trying to answer a scientific question, it doesn't matter from where you start your undergraduates, but if you have to be ready to actually take the challenges for learn something. For example, many of my students who have background from the biology, but they're doing a beautiful amount of math, and I'm really see that how can they do that. So I think mental challenge from it has to be go out. It doesn't matter what is your background, but you can always go to the space biology or any space related thing.
Thank you. That was a great initial comments. So here's my next question for the panel. Now that we are seeing that there's a lot of work that has taken place in low Earth orbit with the International Space Station and several of those efforts, and now we are moving towards understanding how do the environments in lunar orbit or heliocentric orbits, how do they affect living organisms? So I think the question is in two parts. One is, how much of work is still remaining to be done in low Earth orbit that would justify a continued presence, of astronauts within low orbit, or even autonomous experiments to do that in our own close proximity, close to Earth? And at what point of time do we say that, okay, now we have understood our organisms well enough, we've understood how we are well enough in low Earth orbit, and we should now start focusing our energies towards moving slightly away, getting a little independent of our planet and doing some of these studies in lunar orbit, L2 point, Lagrange point, or even further on in Mars orbit, conducting space biology experiments. So perhaps some very quick thoughts on continuing research in low Earth orbit around Earth, as well as moving further away. So how important is it, and how do you see those two different aspects? Dr. Afshin.
All right. So if we go at the pace at which we have been in the past, I see another 50 years for International Space Station. But really, I know that the lifetime is not that long for the International Space Station, and there's so much to learn. We've only just begun to scratch the surface of understanding what happens to human physiology. With the twin studies that was done, even if you look closely at the study, there are so many unanswered questions. It was not even a well-controlled study, even though it's like one of the best studies that we have right now. There's still flaws in there. So in order to, again, going back to the previous thing, we really need to join forces. We really need to come together as nations coming together and answering some of the bigger questions, maybe taking more challenging experiments, putting our resources together to the International Space Station if we really want to become fast-paced about understanding what are some of those physiological changes taking place in human astronauts, so that we can move further on to Moon and Mars. So yes, we really need to pick up that pace. Otherwise, we won't really understand, or we won't have enough answers in good time to go on to further explore Mars.
Thank you. Dr. Ravi?
Hello?
Please go ahead.
Yeah, so I think Afshin is pointed out about the Scott Kelly and Mark Kelly experiment, right? So we know that we understood, but the good news from that study is actually many of those issues we are facing, right? We understood, around 30-40 years we spent using these model organisms for understanding the human health, and many of those studies, especially from the Scott Kelly experiment, that clearly shows that many of those issues we are facing on the space is reversible and preventable by nutritional intervention or pharmaceutical intervention. We can do that as long as we are confined within the habitable space, right? So that I think fairly we understood. in the International Space Station. Probably it might take another, as he said, 10, 15 years to much about this information. But I think one more thing needs to be understood here is space agriculture. Okay, the point I'm making here is we are just trying to understand the growing couple of vegetables that can be used as a supplementary diet for the astronauts. But we are not really into the making these plants as a bioregenerative system that can holistic oxygen, carbon, they reduce the carbon dioxide level and all that. So that needs to be pushed research on the International Space Station, especially using plants as a bioregenerative system. And I think we need to, next few years, it's going to be focusing on the space agriculture, I guess. That will be very, very helpful for long-term space exploration missions because we cannot carry that much of food, right? So that's one area needs to be focused.
Yeah, I think those are all good points. And yep. Can you hear me?
Yeah.
Can you hear me now? All right, I was just saying that I think those are all good points. And what I would add is that we should just go beyond LEO now. We should continue doing research on LEO as long as we can, because as I mentioned, after 3,500 million years of evolution, we're finally able to treat gravity as an independent variable. And we're barely scratching the surface, as Afshin said. There's so much more to learn, so much that we don't know, right? And going beyond LEO is because we do need to have access to that deep space radiation environment that we do not have while we are underneath the Van Allen belts. Our magnetosphere protects us from that. Not completely, but mostly. And in fact, we're trying to get our feet started in terms of that. We're sending a yeast experiment around the moon on Artemis 1. And it's the same thing. We're flying a deletion series and an overexpression series because we want to characterize how the deep space radiation environment affects DNA. We're looking into DNA repair mechanisms, but also doing a correlation between which genes confer increased survivability to yeast, which has over 70% of its most important genes have homologs in the human body. So it's something that was mentioned before with fruit fly, in that it's a really good model organism. You can learn a lot from them, although they're not humans. There's a lot that they can help us understand. So in summary, I'd say let's keep doing research as long as we can in every space station, every spacecraft that we have access to as scientists, but also we need to start understanding what happens in the deep space radiation environment.
Yeah,
Dr. Chandraman?
Yeah, I think those are the very important points. I just like to just add on that what is important, because in the space, you cannot do a lot of experiments, right? Because the way we do in other biology, like the cancer or any drug discovery, antibiotics, whatever experiments we can do, many experiments. So there's a lot, we have a lot of data for them. But now if you want to send one astronaut to the Mars, and now you have to predict from a very small amount of the experiments that how actually all this problem. So there is a huge amount of opportunities for the mathematics and statistics, so that you can actually, whatever the small data we have, from this small data, you have to get a very robust prediction so that we can actually send the people to the Mars. So that is one point. And the second point, in the Lewis way, I would say that the actual fundamental understanding that how microgravity actually affects in the cellular level, because microgravity was never the part of the Earth's evolution. But we do know there is a lot of phenotypic changes at the cellular level in the microgravity, which basically means that when you put the organism in the microgravity, the organism sends something else in terms of some other physical forces, chemical forces, not the microgravity, because microgravity is not the part of the evolution, and it responds based on that. So we have to understand that exactly how microgravity works, specifically for the small organisms like the bacteria, because if you calculate the microgravity effect on the bacteria theoretically, the force is so low, it is lower than the hydrogen bond. So we really don't know that how microgravity is affecting those. So those kind of proper experiments is also very important as fundamental understanding.
Thank you very much. I think with that, I'll pass it on to Dr. Srijay Lakshmi at IIST. Ma'am, please share your thoughts, as well as let's take some of the questions from the students as well. Over to you, ma'am.
Yeah, so thank you so much. I hope I'm audible to all.
Yes, ma'am, we can hear you.
Okay. So in fact, I'm so happy and I'm so glad that all the speakers have done justice to their roles as speakers, and the proof is that they have ignited the minds, because we have lots and lots of questions on the chat box, and our IIST alumni people are so smart that they have compiled all the questions and they have actually sent it to me. So in fact, we do have several challenges, but at the same time, I personally believe that country is actually rich in knowledge as well as rich in the capable people who can carry this work ahead. So along with international collaborations, along with the desire for knowledge, definitely our country will be scaling more heights in this space life sciences regime. So with that, I just wanted not to be in between you and the students, because they are actually curious to know some of the answers from you experts. Okay, so I'm just proceeding with some hand-picked questions, but for those questions which are not picked up, no worries at all. Our IIST Association actually will be posting the questions along with the answers provided by the experts, and they will be available for you for your perusals. So please don't worry, students. Even if your question is not getting selected for the want of time, we have to restrict it to very minimum. But nonetheless, we will be having. I mean, you had already very good sessions, and you can always keep in touch with the experts. They are very nice, and they'll be happy to help you. So with that, I'm just proceeding with maybe a few questions. So I'll go with the first one, because it started with Abibsa Boss. So the question is, will the session involve prospects for biologists to take up astrobiology? I think Abibsa, that question has been already, answered already. So I think experts may not be required here to interfere. I suppose I can go to the next question. So here, there is one more question asked by Abhishek. Lots of questions I can see from Abhishek. So here the question is, he would like to ask about technologies deployment, such as nanosat in space for space exploration during Gaganyaan project from ISRO. I wish if some of the experts would like to comment on that. Siddharth, would you like to?
Lakshmi?
Yes.
Being insider of ISRO, you should be knowing more about this. Actually, this question.
Exactly. In fact, such missions. In fact, nanosat and all is already ongoing missions. I think more aspects should be provided from the ISRO side, because IIST as academic institute, we know such things are going on. But from a mission point of view, I think we should not interfere to such matters. I think we can discuss in some other forums. Okay, so yeah, there is one question. Any research projects or internships available in the field of astrobiology for Indian undergrad students? If yes, where and how? I think Afshin was telling about something on internships available. Dr.
Yes.
Afshin?
Yes. So that would be, it's a very limited thing, because we do this with Blue Marble Space, and we hire, our affiliates hire students from all over the world. So this is something only over the summer. It runs for 4 months, and it's primarily online. Actually, one of my interns from India, I think he's in the audience. I saw Joel there. He's a student at Amity. So Siddharth may have more opportunities probably starting at Amity, because astrobiology just started off there. But then international internships, I know that the German Aerospace Center, DLR, has internships through the DAD Fellowship in India. That's something that's a treaty between Germany and India. And I have met some Indian interns who were studying at Indian universities, and they had gone there over the summer to work at the German Aerospace Center. I'm not aware if NASA gives a paid internship to international students. I know there are ways to collaborate through, if there's like a university working with NASA, they can send their students to work on short-term internships. But again, something that maybe Siddharth or one of the other panelists may know better.
Yeah, thanks.
So to add to that, I think IIST will also have such internship programs. So more if students are more interested to know, they can simply send a mail to our Alumni Association or to IIST direct, so that we can actually have more clarity on that. Okay. Then I'm moving to another question. These studies.
Yes. Yeah, sure. As I already said, like this, the mathematics, statistics, and how the analyzing of the data has a huge amount of effect on the space biology. Because, as I say, first is the microgravity is very low perturbation in one way, and another way we don't have a lot of the data. So you have to develop a new way so that from those data you can actually make a robust mathematical plan which is highly predictive. So there is a huge amount of terms in terms of the bioinformatics, systems biology, and those, right? So definitely.
Okay. So moving on to the next question, target stream and conditions. So can we approach it through astrobiology?
Tardigrades that were sent up in the International Space Station and also I think around a moon mission, these were payloads, and this is not something that Planetary Protection worries about. So I can answer. I had seen this question in the chat box. Or what other kind of organisms are you isolating from spacecraft? So these are all
I think Dr. Ashwini, if you can put, if you can put answers to all those questions collectively, and maybe our Alumni Association will contact you and they will post it, so the students will benefit. Not only the present attending students, maybe the whoever is interested in learning such things also will benefit. Okay, so another question, can plant breeding or plant genetics be done? Are there any scope for a startup in the field of space exploration? I think Dr. Ravi can answer that from a plant.
Yeah, so there is a lot of work has been done on the plant biology experiments, not specifically on the plant genetics. Of course, they have seen that mutations in particular genes, and that could be utilized for the crop improvement programs. Especially there is Chinese research, Chinese space they have been doing the space breeding program, where basically they're exposing to the outer space environment and bringing them back and looking at the mutations rate, because the way cosmic radiations that will induce mutation is very different than what we do radiation studies on the ground. So the genetic studies, of course, you can do that, and there is a huge scope for the space breeding program, and just expose to the space environment and bring them back, look at the screening kind of studies, like look at the plant phenotypes, which are probably going to give the more yield or crop resistance against disease or pest, and all that kind of studies you can do it actually. And there's a good scope for the startup as well for exploring such kind of experiments. And in fact, you can also use the hypergravity studies to screen out some of these phenotypes, which we are doing in our lab currently at University of Applied Science, Harvard. So those are some of the areas you can always explore in terms of crop breeding point of view.
So we are close to the same condition as IIST. You comment on that? Can you hear me, Dr. Louis?
Perfectly. Yeah, I'll be happy to answer. There are different devices that can help you simulate some aspects of microgravity, not all of them. It doesn't matter if you stand on your head and do somersaults. As long as you're on Earth, you're going to be affected by 1G. However, by clino-rotating, so one of them is a clinostat, another type is a rotating wall vessel, or RWV. Another type is a random positioning machine, RPM. Specifically, what we use in our labs are clinostats. And what they do is they are continuously reorienting a cylindrical vessel. So if you have a cell, for example, a bacterium, as soon as it starts sedimenting, it's already on the bottom side. And as soon as it starts sedimenting on the other way back, it's already up. So what you do is you calculate, and this goes back to the question of if somebody studies math, for example, how can they work with this? what we do is we calculate the velocity, the angular velocity with which we have to clinorotate to make sure that that cell, or whatever it is we're studying, stays within a very small zone. We call it a depletion zone. And with that, you can replicate some aspects, not all of them, of the extracellular quiescent environment that is hallmark of microgravity, and basically it just hinders mass transport.
Thank you for that. So one last question, I think with that, I think we wind up. It is to Dr. Siddharth, specifically it is mentioned to you. So somebody is asking, the space biology and astrobiology are two different fields of study in US. You mentioned in your talk, I suppose. Can you please throw some light over that? Dr. Siddharth?
Yes, ma'am.
Yeah.
Can you hear me?
Yeah.
Okay.
Please
Okay.
go
So there are several definitions. Space biology is primarily us trying to understand how a living organism is behaving in space. I mean, I could be wrong. I've got too many space biologists here to correct me. But in essence, that's what it really is. You're taking the living organism into space and you're seeing what happens to it. That could be a human, it could be a fruit fly, it could be a bacteria, right? And astrobiology, in my view, is a bit of that, but it's also trying to understand the larger picture, that where did this life come from? How did it start? How did it evolve? Just like Luis showed us that nice long diagram of how life started on Earth and the oxygen and all of those things. So it's a much more larger, complex question. And space biology tends to be more human-centric, where we're trying to understand, the end goal is to understand what the effect is on humans, whereas astrobiology is a larger question, where it's not only based on what humans are going to be doing, it's about life in general. So I think that would be one way of defining it amongst them.
Okay. So still a lot more are actually there, but I think for the want of time, I would like to pause here. So in summary, I would say that this whole effort from IIST Alumni Association was actually so timely to, and definitely it is going to be a timeless effort as well in the coming days. So I think I can wind up here. So I'm sorry, youngsters whose questions I couldn't pick up for want of time only, but at the same time, we are so happy that we had a very bright panel of members here. So they will be actually answering your queries, and IIST Alumni Association will be posting in their social websites. So over to you, the alumni people, so to take over the function further and to wind up the session. Thank you so much.
Thank you everyone. I think that brings us to the close of this entertaining and very informative session. First of all, I would like to thank Dr. Dadwal, Director IIST. Many thanks, sir, for the inaugural address and for making us capable enough to, being able to organize this session. Next, I would like to thank all the panelists, especially Dr. Siddharth from Amity Center of Excellence in Astrobiology, who has been very helpful in coordinating the efforts to get a diverse and very well-formed panel. Thank you, Dr. Siddharth. Thank you, Dr. Sangram from Saha Institute of Nuclear Physics, Kolkata. Thank you, Dr. Lewis. Very entertaining talk and very informative. Thank you, Dr. Afshin. Thank you, Dr. Ravi Kumar Husmani. And we could not have Dr. Rafael today due to some unforeseen circumstances, but definitely for the benefit of all those who are still in attendance, we are planning to do individual sessions based on the availability of the panelists in future, where there would be sufficient time to tackle all the questions, and maybe we will have much more attendance in that too. So thank you, Dr. Shija Lakshmi ma'am, once again for taking up that Q&A panel. And thank you, all the participants. We look forward for many more such sessions in future, and we look forward to seeing you in attendance. Thank you, everyone.
And please tell us what kind of topics you'd like to cover in the future sessions. We await your feedback. Thank you, everybody.
Yeah, the feedback form has been shared in the chat panel. Please, before leaving, do fill it out so that we can carry on with such more exciting events in future. Thank you, everyone.
Thank you.
Thank you, everybody. Take care.
Thank you, everyone. Thank you, Madhav, for coordinating with Siddharth and the entire team of panelists. Thank you so much. We hope to see you again sometime in the future with many more exciting sessions. Thank you. Have a good night and a good day to Mr. Ravi.
Thank you. Bye-bye.